Hi, I'm Julie Gibson Clark, and nope, I am still not a scientist. But 3 years ago, I did something wild. I climbed to the #2 spot in the Rejuvenation Olympics, edging out Brian Johnson and other billionaires spending millions on their longevity protocols. And I did it on, wait for it, $100 to $150 a month, eating lots of vegetables and taking one simple compound supplement called Novos. Now, my Dunedin pace score was 0.65, meaning I was aging at about 2/3 the speed of chronological time. Pretty good, right? Well, then I messed it all up. One supplement, quercetin, threw my biology upside down and sent my pace jumping to 0.77. So everything I'd worked for, thrown off by a single compound. Now, here's what I'm doing. Over the next year, I'm rebuilding my understanding of longevity science from the ground up, and I'm sharing it with you and crafting a smarter, food-first strategies to slow my aging back down. Will it work? I don't know. In episode 26, about a year from now, I'll retest and we'll find out together whether strategic food-based interventions can get me back to that slower pace. This is episode 3. So today we are talking about autophagy, your body's recycling system, the cleanup process that keeps your cells young. So we are covering 2 papers, Madeo and colleagues on caloric restriction mimetics, which are compounds that activate autophagy without fasting, and Mattson and colleagues on intermittent fasting and how fasting itself triggers cellular cleanup. And I am going to show you a spermidine-loaded salad with jammy eggs. That's not as weird as it sounds. Just roll with me on this one. It's actually delicious. I know it sounds a little weird, but it is a caloric restriction mimetic that activates autophagy without requiring you to fast and includes cheese. So both papers are linked in the description. Okay. Now, when I started researching autophagy for this episode, I realized that I only had a vague idea of what it actually was. I mean, I'd heard the word, I knew it had something to do with cellular cleanup, but if you'd asked me to explain the actual mechanism, I would have totally fumbled it. So, before we dive into these research papers, let's take a step back. I found that everything made so much more sense once I actually understood what autophagy is. And so I'm guessing some of you might feel the same way. All right, so the term autophagy literally means self-eating. Okay, that sounds alarming, like your cells are quietly snacking on themselves. Okay, no, this is less cellular cannibalism and more cellular spring cleaning. It's one of the most refined quality control systems your body has. Think of it like this. Every single cell in your body is like a tiny city with workers, infrastructure, and equipment. So over time, things wear out. Proteins get damaged, organelles become dysfunctional, and bits of cellular machinery break down. So autophagy is your city's waste management system, the cleanup crew that identifies what's broken, breaks it down, and then recycles the material. So without it, obviously the trash piles up and the city becomes dysfunctional, and eventually it fails. That's what happens when autophagy declines with age. So here's the process. First, your cells identify something that needs to go. And then second, it forms an autophagosome, a double membrane that wraps around whatever needs recycling, like a trash bag. And then third, this fuses with a lysosome, your cell's digestive system full of enzymes. So fourth, everything gets broken down into its basic building blocks, amino acids, fatty acids, and sugars. And then finally, those building blocks get released back into the cell as recycled materials. It's remarkably efficient. But here's what's important: healthy turnover. Our cells need to remove what's damaged and replace it with what's functional. Autophagy is the mechanism for that turnover. Young cells do this automatically, but as we age, autophagy slows down. Damaged components pile up and it becomes a vicious cycle. Damaged components make the cells less efficient. The less efficient cells are worse at autophagy, which leads to more damage. It's a downward spiral that accelerates with age. But here's the encouraging part. We can intervene. So we can reactivate autophagy through lifestyle, through food and through timing of meals, and through compounds that trigger the right pathways. When autophagy breaks down, the effects ripple throughout your body. In the brain, harmful protein clumps pile up, leading to memory problems and cognitive decline. In your muscles, damaged mitochondria accumulate, causing weakness and fatigue. And metabolism suffers with insulin resistance on the rise. And in the cardiovascular system, inflammation rises and heart disease risk climbs. So this problem isn't just in one area, it's systemic. And here's the key. When autophagy fails, it triggers a chain reaction. One hallmark of aging triggering another, each amplifying the next. So this ties directly back to episode 1, the hallmarks of aging. Remember, autophagy appears in multiple places. Loss of proteostasis happens when recycling slows down. Mitochondrial dysfunction is connected because mitophagy, the cleanup of mitochondria we discussed in the last episode, is a branch of autophagy. And then cellular senescence arises when the cleanup system falls behind, and chronic inflammation ramps up when cellular debris lingers. So these hallmarks are interconnected, and autophagy sits at the center. When autophagy weakens, multiple aging processes accelerate together. Okay, so you're asking, how do we turn it back on? Several triggers. Fasting and nutrient deprivation. You heard me correct, nutrient deprivation. Exercise, certain food compounds called caloric restriction mimetics, and sleep. Cells are constantly asking, are we in abundance or scarcity? Are we being challenged? Lifestyle choices send those signals. Okay, so the flip side, constant eating, especially high carb, high sugar meals, keep insulin elevated. mTOR, the nutrient sensing growth switch, stays on, telling your cells to build, not clean. And without a challenge, cells have no reason to maintain quality. So, 2 pathways control autophagy, and we touched on this in episode 1. So mTOR, that growth switch, when nutrients are abundant, mTOR is on, build, grow, autophagy off. Now, AMPK, the energy crisis switch. When nutrients are scarce, AMPK is on, recycled, be efficient, autophagy is on. So fasting flips the switch from mTOR to AMPK, and certain compounds can do the same, and that's the focus of today's paper. All right, one fun fact before we get into the papers. Yoshinori Osumi won the Nobel Peace Prize in 2016 for discovering autophagy mechanisms. 2016, people. So we're still learning how to safely activate it in humans. This is cutting edge, real-time aging research. So why does this matter? All right, why spend time explaining autophagy before we get into the papers? Well, because you need to understand the mechanism to appreciate the intervention. When I tell you that spermidine activates autophagy, you need to know what that means. And spermidine is not what you're probably thinking out there. It is a polyamine, and we'll get into that. But when I tell you that fasting triggers AMPK, you need to understand the pathway. And this isn't trivia. This is the language of your cells. So here is the empowering part. You're not passive in this process. Every meal decision, every fasting period, every workout, these are inputs that your cells respond to. And when you understand the language, you can make informed choices, and you have more control than you think. Okay, now let's look at the 2 approaches. Compounds that mimic caloric restriction through molecular signaling, and intermittent fasting through metabolic switching. Both activate autophagy through different pathways. We're going to start with the compounds. All right, paper number 1. Frank Madeo, Didac Carmona Gutierrez, Sebastian Hofer, and Guido Kromer. I'm so sorry, Guido. This is a very interesting name because you've got the E after it like you're going to have an umlaut. All right, caloric restriction mimetics against age-associated disease, published in the Annual Review of Medicine in 2019. All right, this review paper synthesizes decades of research and asks, can we get the benefits of caloric restriction without actually restricting calories? The authors open with a sobering observation. Life expectancy is rising, but so are age-related diseases. Cardiovascular disease, neurodegeneration, cancer, type 2 diabetes, all increasing. We need interventions that actually work. So here's what we know. One lifestyle intervention has been proven across species to extend lifespan. That's caloric restriction. 20 to 40% calorie reduction maintained long-term produce remarkable results. Extended lifespan, delayed disease, better cardiovascular and metabolic health, better brain function, reduced cancer incidence. It's one of the most robust findings in aging research. But here's the problem, as you might imagine. Lifelong caloric restriction is miserable. And for most humans, I mean, who wants to do that? Who wants to be hungry for 40 years? And most people really can't do it consistently. So the question becomes, what if we could identify the mechanisms and activate them through other means? And that's what this paper explores. Now, the authors break down what happens at the cellular level during caloric restriction. Autophagy activation, cells turn on the recycling systems to become more efficient. Then they have reduced protein acetylation, and this affects gene expression and metabolism. Lower acetylation equals better autophagy. And then modulation of nutrient sensing pathways. mTOR turns down, AMPK turns up. All right. Improved mitochondrial function, better energy production, less oxidative stress, reduced inflammation, less chronic inflammaging, better proteostasis, protein quality control improves. All these mechanisms work together. So you touch one, you affect the others. We've heard this a lot. And here's the key insight. If we understand these mechanisms, maybe we can activate them directly without requiring calorie restriction. So the authors propose caloric restriction mimetics, or CRMs. Their definition: compounds that ignite the protective pathways of caloric restriction by promoting autophagy and reducing protein acetylation. Translation: these are molecules that make your cells behave as if you're restricting calories even when you're not. AMPK turns on, mTOR turns down, autophagy kicks in. But it happens through molecular signaling, not energy deprivation. It's biochemical mimicry. And today I want to focus on spermidine because it has the most direct connection to autophagy. It's found in common foods and the evidence is compelling. Now, spermidine, unlike what it sounds like, it is a polyamine naturally present in your cells, and your body makes it, and you get it from food. But as you age, spermidine levels decline. Now, why does this matter? Well, because spermidine is one of the most potent autophagy inducers we know of. It activates autophagy genes and triggers cellular cleanup. Now, here's the science. In yeast, worms, and flies, spermidine supplementation extends lifespan. 20 to 30% increases. Now in mice, spermidine improves cardiac function and reduces cardiac hypertrophy and improves cardiovascular health. It protects against neurodegeneration. In Alzheimer's and Parkinson's models, spermidine reduces toxic protein aggregates. Autophagy clears those proteins before they accumulate. Now, metabolic improvements, better glucose handling, improved insulin sensitivity, and protection against obesity and metabolic syndrome. Now, there is some human evidence. One large epidemiological study tracked over 800 people for 20 years. Those with higher dietary spermidine intake had significantly lower cardiovascular mortality and all-cause mortality. This is correlation, not causation, but the association is strong. Now, smaller intervention trials gave older adults spermidine supplements for 3 months. Results showed trends toward improved cognition and cardiovascular health. And another trial showed modest improvements in vascular function. These are early stage trials, and they are small, short term, but the signal is consistent with the animal models and mechanistically sound. So this is the exciting part. Let's look at some food sources. I can't wait to tell you guys this. All right, so for cheese lovers out there, the richest sources are aged cheeses, aged cheddar, Parmesan, Gruyere, Manchego. The longer the aging, the higher the spermidine. Who does not hanker for a hunk of cheese? Mushrooms, especially shiitake and oyster mushrooms, legumes like green peas, chickpeas, lentils, and soybeans, nuts and seeds like hazelnuts, walnuts, and pistachios, whole grains, especially wheat germ, and then also oats. Fermented foods like natto are incredibly rich, but not readily available, and it's not the greatest taste. But typical Western diet provides about 5 to 10 milligrams daily. Now, studies showing benefits usually use 10 to 20 milligrams or more, but that's achievable through food. A serving of aged cheese, mushrooms, and legumes, boom, you're done. Spermidine affects multiple systems. 1, your cardiovascular, improved heart function, better vascular health, and lower mortality. Neurological, protection against protein aggregates and better cognitive function, reduced neuroinflammation. Metabolic, improved insulin sensitivity and better glucose regulation. The benefits are systemic. Autophagy happens in every cell, so activating it affects every tissue and organ. Here's some limitations. The authors acknowledge human trials are limited. Optimal dosing is unclear. CR mimetics might not replicate all effects of caloric restriction. Individual responses vary. Genetics, microbiome, baseline health all matter. Long-term data are scarce. The authors are excited about the potential, but cautious. More research is needed. Sign me up if it involves cheese. All right, so in summary, the scientists identified why caloric restriction works. Spermidine can activate those pathways through molecular signaling. It directly activates autophagy, and it's found in common foods, and early human data are encouraging. But it's not magic. It's a tool, a practical food-based tool for activating cellular cleanup. And you can start using it today. Aged cheese, mushrooms, legumes, and nuts available at any grocery store. Now, let's look at the behavioral route, fasting itself. Okay, this is our second paper. Mattson and colleagues, Mark Mattson, Walter Longo, and Michelle Harvey. Now, you may remember Walter Longo from episode 1. He developed the Prolon fasting mimicking diet. Okay, so this paper is called The Impact of Intermittent Fasting on Health and Disease Processes, published in Aging Research Reviews in 2018. Okay, so this review shows us that behavior, the simple act of fasting, can trigger cellular cleanup through actual nutrient scarcity. Same destination as spermidine, different pathways. So they define the types of fasting. We've got intermittent fasting, which covers several approaches. Alternate day fasting, eating normally one day, fast the next, extreme but used in research. Periodic fasting, multi-day fast done occasionally, like the fasting mimicking diet in episode 1. And then we've got time-restricted feeding, the most sustainable. Limit your eating to an 8-hour window. Fast the remaining 16 plus hours. You're essentially skipping breakfast or dinner. No special foods, no calorie counting, and you just don't eat outside that window. Okay, Important note here though, before we go further. Many human IF studies are confounded by weight loss. When people do IF, they often lose weight, and weight loss itself has benefits. So when a study claims IF improved these markers, you have to ask, is it the fasting pattern or simply the weight loss? Some studies comparing IF to continuous calorie restriction with matched weight loss suggest IF has unique benefits, but more data is needed. All right, here's the central concept, the metabolic switch. Please understand this. In the fed state, your body uses glucose for energy. Insulin is high, mTOR is on, autophagy is low. But after 12 to 16 hours without food, glycogen depletes, insulin drops, and your body switches fuel source. It breaks down fat into ketone bodies, and ketones become your primary fuel, especially for the brain. This is the metabolic switch from glucose to ketones, growth mode to efficiency mode, and this triggers all the beneficial changes. So when you switch to ketones, you create a cascade of changes. AMPK activates, signaling efficiency mode. mTOR inhibits, turning off growth mode and shifting into maintenance. Autophagy activates, and your cellular cleanup kicks in. Mitochondrial health improves, and you may have more efficient energy production. DNA repair systems also activate, and oxidative stress resistance increases. So on the metabolic side, insulin levels are dropping, insulin sensitivity improves, fat burning increases, glucose regulation improves, and inflammation decreases. These changes carry over and your metabolism becomes more flexible. Now, why this works. So for most of human evolution, food wasn't constantly available. Hello, like no grocery stores way back when. Organisms that survived could efficiently use stored energy during scarcity and activate repair systems. These pathways are survival mechanisms. But the problem in today's world, we never activate them. We eat constantly. We are in the fed state 24/7. Our bodies never get the signal to switch to efficiency mode. We're living in perpetual growth mode, never maintenance mode, and the consequences accumulate. All right, so here's the evidence. Animal research shows robust effects. Even when total calories are matched, the fasting pattern matters. Weight reduction, improved metabolic markers, protection against obesity, diabetes, cardiovascular disease, neurodegenerative disease, and cancer. Health span consistently improves. Now, human evidence is newer but growing. Studies show 3 to 7% body weight reduction, improved insulin and glucose regulation, decreased blood pressure, improved cholesterol, reduced inflammation. These are clinically relevant changes. Now, for brain health, some studies showed improved memory and better cognitive scores. The mechanisms support this. Ketones are excellent brain fuel. BDNF increases with fasting, and autophagy clears the protein aggregates. Now, fasting may have some disease applications. For metabolic diseases like type 2 diabetes, fasting improves glucose regulation and gives pancreatic cells a break to recover. For cardiovascular disease, intermittent fasting reduces blood pressure, cholesterol, triglycerides, visceral fat, and inflammation. For neurodegenerative diseases, enhanced autophagy clears protein aggregates. Improved mitochondrial function provides energy for neurons, and ketones provide an alternative fuel. And then reduced inflammation calms down that chronic inflammation. Okay, so fasting isn't for everyone. It may benefit overweight individuals or those with metabolic syndrome under medical supervision, or people at cardiovascular risk, or healthy adults for prevention. But it's not appropriate for everyone. Underweight individuals, pregnant or breastfeeding women, people with eating disorders, children and adolescents, elderly or frail individuals, and people with certain medical conditions. There is also some new evidence coming out about women particularly and fasting, and we'll have to explore that in a future episode. I will simply put some resources in the show notes to a Dr. Stacy Sims, who's been talking a little bit more about this. It's very new information to me, and I didn't include it here, but I do want you to make note of it. So, critically, we must understand that what you eat during the eating window matters. If you fast, then eat junk, you'll get minimal benefit. Like, hello, right? Fasting activates repair systems. Repair systems require building blocks: healthy protein, healthy fats, vitamins, minerals, antioxidants, and fiber. So fasting signals cleanup. Food provides materials for rebuilding. Both are necessary. So summary, intermittent fasting activates autophagy through the metabolic switch. And when you switch to ketones, AMPK turns on, mTOR turns down, autophagy activates. Evidence in animals is robust. Human evidence is promising but growing. And this works through a different pathway than spermidine, behavioral versus biochemical. But both arrive at autophagy activation. And for appropriate individuals, it's accessible, free, and potentially powerful. Okay, so let's connect the two papers. Paper 1, Madeo, showed us the biochemical route. Spermidine activates autophagy genes through molecular signaling. You can eat your way to autophagy activation. Aged cheese, people, mushrooms, legumes, and nuts, accessible and sustainable. No problem. Now, paper number 2, Mattson, showed us the behavioral route. Fasting activates autophagy through metabolic switching. When you fast, you switch from glucose to ketones, AMPK on, mTOR off. Time-based intervention, also accessible, also sustainable, also free. Hello. 2 different mechanisms, both effective. So this gives you options. Can't fast? Use spermidine foods. Don't want to rely on specific foods? Use time-restricted eating. Want maximum benefit? Combine them. That's what I am doing. So fast for 14 to 16 hours or whatever your fast is overnight, and then break your fast with spermidine-rich meal. Fasting activated autophagy through metabolic switching, and then spermidine continues activation through molecular signaling even when you're eating. Both pathways work simultaneously, and that's what today's recipe does. It's designed to break a fast while continuing autophagy activation, the best of both worlds. So both approaches are remarkably accessible. Fasting is free. Spermidine is in common foods at any grocery store. These aren't expensive supplements or prescription drugs. This is budget-friendly longevity. Now, some people can't fast, and spermidine offers an alternative through food, not deprivation. And not everyone needs to fast to support autophagy. So now you have multiple tools: time-restricted eating, periodic fasting done occasionally, spermidine-rich foods eaten regularly, and exercise that also activates autophagy. Choose the way that works for your life, health status, and preferences. There is no single right way. The right way is the way that you can sustain. Now, let's make this practical and delicious with a spermidine-loaded salad. So this is a spermidine-loaded autophagy salad with jammy eggs. Every ingredient is chosen for autophagy activating potential, and this meal delivers over 100 milligrams of spermidine from whole foods. Now, for context, human studies used about 6 milligrams per day, so this provides 15 to 20 times that from ingredients that you can buy at any grocery store. So it's designed to break a fast, but it doesn't require fasting. The spermidine activates autophagy regardless of when you eat it. And that's the beauty of a caloric restriction mimetic. Let's go through our ingredients. So I've got about 3 ounces of aged cheese here. I've got a mix of Parmesan and Manchego, and this is going to deliver about 70 to 80 milligrams of spermidine. This is your MVP. Aged Manchego, Parmesan, cheddar, Gouda. Look for something that's 18 months or older minimum. And if you're at Costco, the Manchego there, that's only about 3 or 6 months. But the Parmigiano Reggiano is 24 or 36. Great place to pick that up. Then I've got 2 cups of mushrooms. That's going to give us about 15 to 18 milligrams of spermidine. Shiitake is your priority, but cremini, maitake, and oyster all work. Then we've got 1.5 cups of peas. That's going to be about 9 to 12 milligrams, fresh or frozen. I have frozen here. Then I've got more than a cup, but a cup of crispy chickpeas. That's another 5 to 8 milligrams of spermidine, and that gives us a nice texture context. Now back here, I've got some hopefully jammy eggs, which is going to give us some protein, some choline, and some richness. I've also got some walnuts. That's also going to have spermidine and omega-3 fatty acids, plus elagitannins, like we talked about in the last episode. And I've toasted up about 2 tablespoons of wheat germ, also giving us another 15 milligrams of spermidine. Skip it if you're gluten-free. Then we've got arugula. So this has got all our sulforaphane precursors, and it's got a nice peppery bite. And then in here, you don't see it, but I've got olive oil. We are going to be using balsamic vinegar, a little bit of parsley. So those all provide some polyphenols and some brightness. So if you add this all up, you should be getting anywhere between 80 and 120 milligrams of spermidine. How cool is that? So here's how we're going to make the salad. It's pretty easy. I've already prepared these jammy eggs, and I'll talk about that in just a second. But then you need to toast your walnuts in a dry skillet. Super easy. And then if you have spermidine, you're going to want to also toast that too. It brings out this really bright, sweet flavor in the wheat germ. Excuse me. And then to make these crispy chickpeas, I've put the recipe in the show notes. It could not be easier. You just toss them in some olive oil and some spices and pop them in the oven. You can make them in a pan as well. I just like doing them in the oven because it's just less faffing about, but it's super easy. I will have that recipe in the show notes, and that makes a great snack, especially if you're a crunchy kind of snacker like I am. So the very first thing we need to do is saute our mushrooms. So we're going to saute the mushrooms in olive oil and a little bit of butter. So 2 tablespoons of olive oil and a tablespoon of butter. It's going to be delicious. We're going to saute those up, add a little bit of thyme, and then we're going to add just a tiny splash of white wine at the end. We'll pull that out, and then we're going to put the peas in there, and then we're going to saute those up with some garlic as well. And then we are going to assemble our salad. Now, I've already made a quick balsamic dressing. I will put the recipe in the show notes. So easy, but it's basically balsamic vinegar, olive oil, and Dijon mustard. It's that simple, and it just brings this whole thing together beautifully. So all of that will be in the show notes. Why don't we get started, and we will saute our mushrooms, and then I'll tell you what I was doing back here with the eggs. I'm experimenting with my little egg cooker. But what you'd really like with the eggs is you want like a jammy egg, and if you don't have one of these cookers, no big deal. You just cook it in boiling water for about 5 to 6 minutes, and then you can put it in an ice bath, probably closer to 6 minutes. And then you want to put it in an ice bath and stop the cooking. And that hopefully will give you this nice, that jammy egg where the yolk isn't completely cooked, but it's not super runny either. First, we're going to turn on our skillet here, get that heating up. Now, the thing with mushrooms is you never want to crowd the pan. So we're going to have to probably cook these in batches because I don't have a very big pan, but it's going to be delicious. Oh, it Sorry, I sing when I'm happy, and when food is going to be super good, I get very, very happy. I'm so excited about this. So while we're waiting, let me just see. What I did is, okay, I put these on the egg cooker and said, okay, cook it to soft boiled egg. And then I put one in an ice bath. You can see I put IB on it. And then I put one I didn't. And I wanted to see if I stopped the cooking versus not stopping the cooking, how different those would look. So I'm going to peel them now, and we'll I'll set them on each side here, and we'll see what happens at the end. I have a feeling they're not going to produce much jamminess, but we'll see. We'll see. I love this little cooker because it's super, super convenient. Ooh, I don't know, maybe they are jammy. We'll see, it seems quite soft. So let's, yeah, that's probably ready. So I'm gonna put this down, and we'll do more of this in a second. Oh dear. You have to be careful when the eggs are soft boiled, unlike hard boiled. You know, you have to be a little bit more careful when you peel them. Okay, I'm gonna put my oil in. I'm multitasking, which is a bad idea. Okay, yeah, that's 2 tablespoons of olive oil and a tablespoon of butter. Oh, you don't have to use the butter. You can just use olive oil if you'd like. But we'll let that melt. I'm gonna get out a bowl. So we're gonna do this in batches. Okay, so again, don't crowd the pan. That's probably plenty. And then we'll do a second batch. So while that's cooking, I'll finish peeling my eggs, oh so carefully. Now these have gone a little bit cold. Ideally, they would just be a little bit warm because the whole thing comes together beautifully. I'm just gonna set it on our greens. So that one is going to be our ice bath, and this is our non-ice bath. And I am going to put this, I got to figure out a place I'm going to put this so that I know which is which. I wanted to do this test and see. If you've never used one of those, again, I'm not trying to sell you this thing, trust me. I thought it's like 20 bucks on Amazon. I think my mom even gave me that, but it's so handy. I'm not big on having a lot of stuff in the kitchen, mainly because I have so many tools and, you know, things. I always think, oh God, I don't need one more thing. But that and the Instant Pot was one of those, like, oh, I don't need one more thing. And then I've just been so pleased that I have them. So I don't know what it's worth, but I thought I'm going to try to figure out how to make the jammy eggs in there because I haven't cracked the code, so to say. Sorry. Okay, that's really cheesy, isn't it? All right. Let's see how our mushrooms are sauteing. And because I'm going to add a splash of wine, what I'm going to do is I'm going to cook this. I'm going to pull it out, then I'm going to cook the other batch, and then I'm going to put them all back in and just do a quick splash of wine and let it all marinate in that wine together. Okay. These are almost done. We want to get these nice and caramelized. And mushrooms are something, one of the things I've learned in the few cooking classes I've taken or been a part of, they can really kind of take a beating. You can saute these things and they can kind of go a little while, similar to onions. So you can not overcook. You can't really overcook them. You can kind of let them be in here for a while and they can kind of take a beating. So I'm going to let that go. We'll come back here in a little bit when they have caramelized a little bit more. Okay, so these are, the second batch is cooking up nicely and they've kind of shrunk in size. So I'm going to put the other batch back in. And we're going to let these caramelize together. I'm going to put some thyme in here. I've got some fresh thyme. The jokes are never Never end with the spice time, huh? If only. So you need about a half a teaspoon of dried thyme. So I'm going to use a little bit more than that of the fresh thyme. Okay. Let that flavor kind of bloom together. Thyme and mushrooms are a wonderful flavor combination. Anytime you want to have sauteed mushrooms, try not to forget the thyme. That adds a really beautiful hint of something else to it. I'm just going to do a nice little, not all of this wine, so probably about a tablespoon of white wine, and then we'll just let that all evaporate off. And that just adds that nice little extra to it. I'm just going to get all that wine to evaporate. Oh, it smells so good in here. Okay, so shiitake mushrooms, we're going to put them back in this bowl for right now. And then the beauty of this, so I use this as a dry skillet to toast my walnuts and my wheat germ. And then you put the oil in and you're going to use it for your mushrooms. And now this has got quite a bit of oil, but we're going to use it for our peas as well and our garlic. So garlic and peas into the pan. How nice is that? So it's somewhat of a one-pan dinner if you have everything else ready. And then we'll saute that up. I'm just going to turn it up just a little bit. Since those peas were frozen, they cooled down the pan. Okay, so our peas are pretty much done. We just want them heated through, and you want to kind of be able to smell the garlic. You don't want your garlic to burn. You can see it's kind of browning nicely, so I'm going to turn it off. And then I'm just going to pull these off, and then we'll assemble the salad. So this salad is a really great lunch if you work out of the house and you need to pack something up, because basically you can get all of these ingredients ready on a weekend or any other time, and you can prepare them in bulk, and you can bring this salad every day if you want, and you just kind of bag up and package up each little ingredient, and you're going to mix it all together. So the first thing we start with as we assemble this salad, let's move this out of the way. Let's move that there. I'll move this a little closer here. Okay, so the first thing we want to do, first of all, we've got our ice bath egg and our non-ice bath egg. I have got some arugula at the base, okay? And then I'm just going to add a few thinly sliced onions. Now, again, if you were bringing this to like work for lunch, you could just, that could be one thing that's packaged together. I just wanted some of that. I thought that would taste really good. And I also want to add some parsley, because I just think, one, it's going to add some nice flavor against the mushrooms and the peas, but also, you know, more polyphenols. So why not? So we'll just do a little bit of that. Just kind of, whoops, mix that in there. Now, we want our mushrooms to still be warm. So again, if you brought this, you kind of want to reheat your mushrooms somehow, but you want these ideally warm so that it kind of wilts the arugula just ever so slightly. So we're going to layer on our mushrooms, and we're going to layer on our peas. Now, truth be told, I'm not sure I'm going to use all of these peas. That's a lot. I think I'm going to save some for later, but we'll try to use quite a bit here. Yeah, a cup and a half might be too much. I don't know what I was thinking, but I think a cup of peas is probably plenty. Then we're going to want to do our crispy chickpeas. Now, again, I said a cup in the recipe. I'm going to adjust it because I think just enough to scatter. So this will probably be about 2/3 of a cup. You want that nice, crisp crunch. I'm going to put a little bit more on. I think that'll be good. So that's a little under a cup. And then again, we're going to do some walnuts. That's got that omega-3 fatty acids and the elagitannins, and then all of this wheat germ. I am going to use all of that because that's my spermidine-loaded ingredient. Now, just try to make this look a little bit prettier. Okay. Okay. So that arugula is like wilting down under the heat of the mushrooms and the peas. Then we are going to nestle these jammy eggs, hopefully jammy eggs, we'll see. It's a work in progress in this kitchen at all times. Not so jammy. So this is the one that I didn't put in an ice bath. Still pretty nice, but I'd like it a little bit jammier. So I'm gonna put that, try to make this as pretty as I can. And this, oh, I can feel this is soft. So this is the one that I put in the ice bath, and it feels a little softer. Oh yeah. It's got some nice jamminess to the yolk, which is really delish when it mixes with the dressing that I'm gonna put on here in just a second. Now I'm just gonna do a little bit of salt all around and a little bit of pepper. Okay, and then I'm gonna do our dressing, and then we're gonna top it with the cheese. Now, I don't know how I'm gonna get all of this cheese on there, so we'll see. We might not be able to do all of that. And in fact, maybe this salad might be better for two people. But so just want, this is again a classic balsamic that's been emulsified together with the Dijon mustard. Super, super simple, okay? So we're just gonna dot this around, and then it's hopefully enough that you get kind of some in each bite. Now, another way to do this is to toss the arugula with the dressing ahead of time and then put that together. But we're just gonna dollop this all over our salad. And then I'm going to attempt to put all this cheese on here. Look at that. I don't know, I think that's kind of good news that cheese, some kind of cheese is good for you, especially as we're going into the winter. All right, this is not working. In my efforts to get as much spermidine in this salad as possible, I don't think it's come together so beautifully. So don't let perfect be the enemy of the good. What we're gonna do here is take off these beautiful jammy eggs. I'm gonna take those off, as unbeautiful as this all looks right now, but guess what we are gonna do? It is gonna be actually so delicious. Hold on one second. So we are gonna toss this all together. It's gonna be beautiful and taste a lot better. Now, cooking is one huge experiment, and this is no different. So this is gonna be so much better all tossed up instead of trying to compose it. I've made it before all tossed up, and I don't know what I was thinking. I thought I could compose it and make it look prettier, but I think it needs to be done like this. So you can kind of see I've got it all tossed up here. Then we're gonna put it in our bowl, and that's gonna be much, much more easy to handle. We get all that spermidine, all the polyphenols, just all the dressing is evenly divided through every bite. Okay, so maybe not completely as beautiful as a composed salad, but still quite lovely. Now we can put our eggs on top, which don't look as pretty, but again, don't let perfect be the enemy of the good. And I hope that if you learn nothing else from me in all these shows, I'm not about perfection, especially when it comes to the kitchen, and it's probably why it's my favorite place to be. So this didn't perfectly come together, but it's still absolutely beautiful, and I cannot wait to eat it. So I'm gonna have a few bites of this, and then we are going to wrap up this episode, and we'll talk about the limitations and cautions as well. I will be right back. Okay, that was delicious. This salad is so good, and I apologize for everything. I was trying to kind of make it look fancier than when I normally do it, because when I normally do it, I do just dump it all in a bowl and mix it all up, and then I put it on in a bowl similar to that. And I just thought I could make it look a little prettier, but it's so delicious. The peas with the mushrooms, with the cheese, the garlic, and like the wine, it's all just something that comes together more beautifully than everything on its own. Now, I'll put the nutrition information in the show notes as well, but I think there's about 40 to 45 grams of protein. If you count that, there's about 100 to 140 grams of spermidine, depending on what you use. So spermidine is an autophagy activator, but I need to be very straight with you. Let's just go back to basics. Exercise is your number one autophagy activator. It's more powerful than spermidine and more powerful than fasting. AMPK activation from exercise triggers autophagy throughout your tissues, not just in your muscles, but everywhere. And listen, walking counts, strength training counts, playing with your kids all count. Movement matters, and it doesn't have to be complicated. Just move to trigger that autophagy. Okay, now, here's the limitations and cautions you probably need to be aware of. Your microbiome, your genetics, and your baseline health all affect your response. Personalization matters, but we don't have clear guidelines on how to do that yet. So you are going to have to decide for yourself. Now, optimal dosing from food is unclear when it comes to spermidine. Individual absorption varies, and long-term effects haven't been studied in controlled trials for years or decades. Now, when it comes to fasting, underweight individuals, pregnant or breastfeeding women, and people with a history of eating disorders, children, elderly or frail individuals, and people with certain medical conditions, fasting isn't appropriate. Always consult your doctor. Now, we're working with normal foods here and Mediterranean populations that consume high amounts of spermidine with excellent health outcomes. Spermidine supplements are different. They're concentrated doses, variable quality, and minimal regulation. So if you're considering supplements, I would also advise you to ask your doctor. Now, I am a health influencer. I am not a doctor, and I'm only sharing like what research and what I've learned and my approach, but your situation is unique. Again, talk to your health care provider for medical decisions. Don't let me be that person. I can't even pass chemistry, people. Don't listen to me for medical advice, okay? But food is a foundation. It's not magic, but these are the tools that work best as part of a comprehensive approach. Whole foods, movement, sleep, stress management, and social connection. You just need to start slowly, track how you feel, and if something is not working, just adjust or stop. Sustainability in this program or eating healthy matters way more than perfection, okay? So, autophagy is your cellular recycling system. It declines with age. And when it does, multiple aging processes accelerate, but we can reactivate it. Spermidine activates autophagy genes through molecular signaling. Found in aged cheeses, it doesn't get any better than that. Mushrooms, legumes, nuts, no fasting required. But intermittent fasting does activate autophagy through metabolic switching, time-based intervention, free and accessible for many. So both arrive at the same destination, and you can use one or both depending on what fits your life. Now, these aren't expensive. Fasting is free. Spermidine foods are at your regular grocery store or maybe a cheesemonger for the aged Manchego. But this is a budget-friendly way to longevity. Real food, strategic timing, evidence-based, and accessible. You're not passive. Every meal is a signal. Every fasting period is a signal, and every movement is a signal. Your cells respond, and now you have more control than you think, and you know that now. Over the next 23 episodes, we'll keep building on this toolkit. And by episode 26, we'll see, did these protocols work for me? Did I get my pace of aging back down? I hope so. You're watching this experiment unfold in real time, and I hope you can see it may or may not work. We don't know. I don't know. We'll find out together, and along the way, we're going to eat a bunch of delicious meals. Every meal is an opportunity, not just for calories and not just for pleasure, though those certainly matter. It's an opportunity for cellular health, an opportunity to activate beneficial pathways. Food is information, not just fuel. In 2 weeks, we've got episode 4, Inflammaging. Inflammation's double edge. We'll explore the difference between acute inflammation that heals you and chronic inflammation that ages you, how it connects to everything we've learned so far, and how to build an anti-inflammatory pantry or kitchen using the research. Comment below. Have you tried intermittent fasting? Are you already eating spermidine-rich foods? What's your favorite aged cheese? I want to hear from you and I want to learn from your experiences. If you're a scientist and I got something wrong, please tell me constructively. Help me learn and help me get this right. I'm here to understand the research accurately and share it fairly with everyone who's watching. Subscribe. Please like and subscribe if you want to follow this 1-year experiment and learn accessible paths to longevity. Episode 26 will reveal whether these food-based protocols work and whether I got my pace of aging back down. You are watching the journey unfold with me. Your cells have a choice every day: accumulate damage or clean house. For most of modern life, we've been signaling them to skip the cleanup. Constant food, no challenge, no scarcity signals. And our cells have been listening. They're building up decades of cellular trash. But now you understand the recycling system and you know how to flip the switch from growth mode to cleanup mode. You know spermidine can activate that through food and fasting can activate it through time. And you have options that fit your lifestyle. So this isn't about perfection. I think you can see that. It's about sending better signals more often. It's about giving your cells what they need to maintain themselves. You're not fighting aging. You're reminding your cells how to take care of themselves. Okay, in 2 weeks, Inflammaging. Why chronic inflammation accelerates every aging process we've discussed, and how to build an anti-inflammatory kitchen. From paper to plate, I'll see you then.